All right. I updated that last lecture, so it's now updated, the Virology 3.4. It's on Moodle. So if you want to re-download it, it's available for you. I just changed like 2 things on a slide, pretty much, but important things on a slide. There's, I think, 2 slides that need to be edited. Some of these slides I inherited, and so I apologize. I didn't look through all of the words. So that was on me, but now it's updated. All right. Moving on. Let's see here. Any questions on viruses before we move into immunology?
I think I missed one part. Are you skipping over the respiratory viruses?
No, we're pausing here. I want to make sure we cover immunology, and then I'll come back to viruses at the end if I don't. And what will happen is if we don't have time to get back to the rest of the viruses, I will record a recording to post for y'all. But I want to make sure that we have at least like 2 hours to talk through immunology before we close out today.
Sweet, thanks for repeating that.
Yeah, no, thank you. I appreciate it. Perfect. So we'll jump into our immunology presentation. And then, like I said, if we don't have time to come back to viruses, I don't want to rush through anything. So I want to make sure we can fully discuss these pieces. And then what I'll do is I'll just post a recording of the tail end of viruses. If we don't get through them today, I'll probably record that like tonight or tomorrow, and then you can have it up for your viewing pleasure if you want to review those respiratory viruses. And then we also get into the gastrointestinal viruses. So it is important content. But this, I think, I want to make sure we talk about as a group. All right. Perfection. Okay. So getting into our immunology reviews. This is our 3.5. And this is going to be a nice reintroduction to our key players of our immune system. And I have it broken down into a couple different sections, so hopefully it'll help us mix things up. But first, just to reorient us to the immune system organs, our anatomy, we have our primary organs, which are our bone marrow and our thymus. The bone marrow is responsible for our immune cell production overall and B cell maturation, versus our thymus is where we see our T cells mature. Our secondary immune organs are going to be the spleen, the lymph nodes, tonsils, Peyer's patches, and really their job is to help our immune cells interact with an antigen. The lymph nodes in particular have a couple different kind of roles and components that we'll see. They have some non-specific filtration that's done by macrophages to identify antigens coming through. They allow for circulation of B and T cells, allowing them to communicate and get exposed to antigens. And really, it's the site of our immune response activation. So looking at the anatomy of the lymph node, they're an encapsulated structure. They have trabeculae. We can kind of see these little trabeculae forming in, so they have these different little spaces. There is the follicle. If we look at the actual, let's see if I can find a good, well, I mean, these are all follicles here. I just don't have anyone that's like perfectly named. But our follicle has an outer cortex portion and a B cell localization proliferation portion. So we could call this the outer cortex of the follicle, and then the B cell zone, essentially, or proliferation portion. The paracortex region is going to be the region kind of surrounding these follicles here, this paracortex region. And this is where our T cells will hang out, is in our paracortex. So in this like blue section here. In DiGeorge syndrome, it's actually underdeveloped, this region. So we see T cells have a harder time engaging with their antigens. And we'll see that it will get, this is the section that gets really big in an extreme cellular response. So a viral infection where you have lymphadenopathy, Epstein-Barr, things like that. This is the section, the paracortex, that really expands out and becomes enlarged. And then we have the medulla in this space here. And the medulla space, we have these medullary cords, which are like really closely packed cells, lymphocytes and plasma cells. And then the medullary sinuses, which are where reticular cells and macrophages hang out. And this is where our like lymph flow will exit from. So we can see lymph flow will enter via our afferents, and lymph flow will exit via our efferents. Efferents for exit.
Quick question.
Yes.
All lymph nodes have multiple entries and a single outflow, or is
Yes.
that variable? Okay.
Yep, they'll have multiple entries and a single outflow, correct? Yeah, excellent. Think about it like this, right? Our lymph nodes are kind of these collecting spaces. And so we're collecting from all the way down in the periphery up until ideally that thoracic duct, that collecting duct up top. And so we're trying to shorten and decrease the amount of lymph pathways that we have to take to get there as we move up the body closer to that big drainage system into our venous system. Awesome. Great. Excellent. But yes, and you'll see that there'll also be communication here, almost in that hilum. It kind of looks like a kidney, right? With blood flow. So we have veins and arteries, and then they will actually communicate, though, with the individual follicles and throughout the lymph node, but they'll enter and exit similarly. Now, I don't want to spend a ton of time on this slide. I really have it here for your reference only, but this is your different types of lymph nodes or locations, what's draining, who's draining what where, and if there's a pathology associated, where might the lymph node be present. And so I have a couple of slides of this, 2 slides of this. Where I see this to be probably the most important would be potentially supraclavicular, because your right is your Virchow's node, your left is typically some type of malignancy of the abdomen or the pelvis. So those ones come up more commonly on an NPLEX exam just because they're unique. We can also obviously see our axillary because of metastasis from breast cancer, so those ones are more unique. Epitrochlear, secondary syphilis is an interesting association with that one in particular. And then our abdominal, we see abdominal pathologies, right? Our inguinal, we see kind of inguinal pathology. So these ones aren't as exciting or or jumping out of the page. But you can, if you really wanted to go to town, you could learn them all. I really focus on some of the main kind of unique ones, or the ones where they have specialized names. Looking at the spleen, left upper quadrant of the abdomen is where our spleen is going to be. It's going to be anterior and lateral to left kidney, so in front of the left kidney, but more lateral to it. There is this periarteriolar lymphatic sheath, meaning it's fibrous and contains vascularity, and it's occurring throughout. That's why the spleen appears kind of red or meaty or like juicy, almost like piece of meat, because there's so much vascularity going to it. In this section, there's T cells that are located within the white pulp. And then in the follicle, you'll see B cells. So you'll see there's this marginal zone here that we can also see. This contains macrophages and specialized B cells, APCs capturing bloodborne antigens to be able to recognize for lymphocytes. We can see our primary follicle, our white pulp, and then our red pulp surrounding where that vascularity really is, where we can see all those different blood vessels. Okay. So anytime we're talking about the cells themselves, that's going to be in our white pulp, versus if you think about the vascularity of the blood vessels, that's going to be in your red pulp. Much more red pulp than white pulp in the spleen. Okay. For if we had a post-splenectomy spleen, you could see things like decreased IgM, decreased complement activation. You could see some complement opsonization issues, increased susceptibility to encapsulated organisms, because if we can't do opsonization, we can't activate complement, our encapsulated organisms are going to roam free. So pneumococci, Hib, meningococci, these essentially bacteria are going to be more likely to infect us. And microscopic findings like Howell Jolly bodies, target cells, thrombocytosis, and lymphocytosis are common findings in a post-splenectomy spleen world. I put this on here because there was a question on the board exam one time regarding this. So this is your, what would happen if the spleen is removed? And if you want a laugh, you can watch What Does the Spleen Do by Harvard Medical School. All right. Our thymus purpose, its anatomy and its function. We know the thymus gland is kind of middle mediastinum, but we also know it starts to go away after kind of we move through adolescence into adulthood. Its purpose is to help T cells differentiate and help T cells mature. We see it develop embryologically from the third pharyngeal pouch, which is the endoderm. That's where the thymus epithelium, the lining of the thymus, occurs. But thymic lymphocytes, the cells, come from mesoderm. So just a little embryology tie there. And if we look at it microscopically, we'll see that there's a cortex region that has immature T cells, an intermedullary region that has mature T cells, and then there's these corpuscles, the Hassall corpuscles, which contain epithelial reticular cells, which are APCs, or communicating cells, helping these cells become mature and exposing them to antigens to help them go through this self-selection process. An interesting pathology of note in this tissue is a thymoma, which is associated with myasthenia gravis. You also can see it being associated with superior vena cava syndrome or pure red cell aplasia or Good syndrome. But myasthenia gravis is the only one of these that's on your NPLEX review sheet. So that's why it's underlined here. So moving on, now that we have kind of the players involved, we want to reintroduce the concepts of innate and adaptive immunity and the cellular components in each. So if we look here, we remember our innate immunity is going to be our hours response to any type of infective organism. It's going to be more generic in nature. And some of the players will be our epithelial barriers, our phagocytes, our complement, and our natural killer cells. Versus adaptive immunity is going to be days after infection. It's going to be our memory, and it's very specific. So our B lymphocytes that eventually produce antibodies, and our T lymphocytes and T effector cells, which we will re-meet all of these players here in a moment. So there's two arms of the immune system. Innate is the differentiation between harmless and harmful impacts to the body. It's also the detector of our stress and danger signals. Our adaptive immune system is the differentiation between self and non-self structures, and it's antigen-specific recognition. So what does this look like? The cells involved, if we look at the cell-mediated components of innate immunity, we have this whole list here, our neutrophils, our macrophages, monocytes, dendritic cells, et cetera, And adaptive immunity has a much smaller list of cellular components like T cells and B cells, and then it has its humoral components of circulating antibodies. So when we look at specifically our innate immune cells and their function, our monocytes or macrophages, we can think of them as being famous for phagocytosis, inflammation creators, T cell activation, and tissue repair. Our neutrophils is going to be phagocytosis as well as inflammation activator. Natural killer cells is going to be our killing infected tumor cell, killing either infected or tumor cells. And our dendritic cells is going to be also phagocytosis and activating naive T cells to kind of induce that adaptive immune cell response. And then lastly, we have our mast cells with help with inflammation by being able to degranulate and release things like histamine. And eosinophils are other granular cell, which allow for us to have some parasitic defense. When we look then at some of these different kind of mechanisms, cellular mechanisms in response to pathogens, our innate immune system is germline encoded and non-specific, rapid with no memory. So that's really what those cell players are doing. Versus adaptive immunity, we'll see variations through VJ or VDJ recombination. So essentially their antigenic recombination during lymphocyte development, allowing them to be antigen specific for different antigens that they may encounter. Over time, this becomes more and more refined as we're exposed to more antigens throughout our life, and we can have long-lasting memory. And these memory responses then, if they occur again a second or third or fourth time, will be much faster and much larger. So this is, again, great for quick detection, great for specific long-term support. When we look at, kind of some of the secreted proteins and pathogen recognition patterns in the innate immune system, proteins that are secreted here could be anything from lysozyme, complement, our C-reactive proteins, defensins, cytokines, things that are not just from immune cells, but also from our epithelial lining. And we'll see on the adaptive immune side, we're really talking about two different languages, which we'll look at, cytokines and immunoglobulins. Versus if we look at our innate immunity when it comes to pathogen recognition, again, they're non-specific. So we're looking at toll-like receptors, which recognize certain patterns in bacteria. This is often called a pathogen-associated molecular pattern, or a PAMP. And they might recognize things like a certain type of receptor, or a certain type of flagella, or a certain type of generic protein, but they aren't necessarily saying, oh, that flagella is associated with this bacterium. And this protein's associated, this catalase enzyme or whatever is associated with this exact bacterium. They're saying, oh, this must be foreign because it has this thing. And so our pathogen recognition in innate immunity is much more generic. Its goal, though, is if we're able to have these toll-like receptors, these TLRs, recognize a pathogen-associated pattern, it'll activate things like our natural killer cells, which then can actually destroy those cells that are now foreign that we've recognized. We can recognize things like LPS in our gram-negatives, flagellin, nucleic acids, etc. Versus on our adaptive immune side, we can be very specific down to the exact antigen on the surface of a virus or a bacterium. And our memory cells, our B and T cells that are activated, are able to do this, and they're able to hold and retain that memory. So now going in a little bit deeper into one of the important players of not only innate immunity, but also activation of adaptive immunity is phagocytes. And phagocytes could be macrophages, but there's a lot of other cells that can also do phagocytosis, if you remember that big list. So I'll often reference macrophages on here a lot, especially if it's a specialized job that they do. But any phagocyte can do some of these pieces of the puzzle. So we look here, we can kind of follow along what's happening in this process. So the pathogen enters into the system, and the pathogen becomes recognized by macrophages and dendritic cells typically first in the innate immune system. This recognition is nonspecific and via those PAMPs, those pathogen-associated patterns that I mentioned. Then once that pathogen is recognized as foreign or as harmful potentially, then we'll undergo phagocytosis and then killing of the pathogen. And this is where macrophages shine, and they can secrete reactive oxygen species, nitric oxide, or lysosomal enzymes to actually kill that pathogen once they've phagocytosed them into a phagosome. And this is that phagosome here. And this is them, killing that phagocytosed microbe with whatever means necessary. Now, I mentioned some microbes are able to either deactivate this process of having these reactive oxygen species or lysozymes being present. They can stop the fusion of these two things here, or they can actually even just get out and escape the phagosome or phagolysosome and get into the cytoplasm. So bacteria are pretty tricky about evading this mechanism, but if they're able to get phagocytosed and are able to actually undergo this process, this is who's doing it, a macrophage, and what they're using to do so. After they phagocytose things, they're then going to essentially display them out, out into their surface, essentially of the cell, saying, hey, I just engulfed and digested this molecule. And that is going to be part of their process of inducting inflammation. And that's really macrophages who's doing that job as well. They'll also produce pro-inflammatory cytokines after that phagocytosis process. And that includes tumor necrosis factor and interleukin-1, as well as other chemokines to bring different molecules here. But when in doubt, think of TNF and IL-1 as your big pro-inflammatory cytokine molecules. This process of these pro-inflammatory molecules and cytokines and these chemokines are going to attract other cells to the infection site, specifically getting things like neutrophils and other innate immune system cells there. And they'll also use adhesive molecules like selectins and integrins and chemokines to get them there and hold them in that place. We can kind of see some of these processes here. This is the blood flow inside of a capillary or blood cell. This is the endothelial lining, and this is showing how leukocytes will arrive at the site of infection and that extravasation process. So our macrophages have secreted TNF alpha and interleukin-1. They're saying, hey, inflammation, warning. We see this upregulation of these fibrin molecules, these selectins, and these cells start to weakly bind to these selectins and roll along the side of the blood vessel. They then, the integrins, they're able to actually bind firmly to an integrin, and it stops the roll, so the cell's not rolling anymore. And then they're able to transmigrate and undergo essentially diapedesis between these endothelial cells and leave the blood and go into the interstitial tissue. Very good, I started recording. And then we're able to escape. I think that was it. Yeah, we're able to escape. And then the last step that it does is after it's eliminated that pathogen, it's going to either fully be gone and then no other, new immune cell response is needed, or oftentimes, and it's going to activate our adaptive immune system. So then dendritic cells, macrophages, and neutrophils all can do a job here in activating that adaptive immune system, and also with pathogen elimination. And then after this is done, after the adaptive immune system is on the job, pathogens are gone, then we'll start to see our phagocytes do tissue repair and remodeling. Macrophages really do that the best. muck up any debris, break it down, digest it, and they'll use enzymes and cytokines like growth factor and metalloproteinases to help kind of clean up the system and repair the tissue back to normal. So phagocytes are really a major player when it comes to our innate immune system and their role to kind of get things going when it comes to attacking something at that localized level. So we're going to look now at some immune cell functions and some of the diseases associated with those immune cell functions. Before we get to those kind of like details, we have to understand some different protein molecules, some different receptor complexes. And I've mentioned these a few times, and they're major histocompatibility complex, MHC. I'm just going to call them MHC from here on out because it's so much easier. But MHC1 loci have one letter, and MHC2 loci have two letters, so hence their names. MHC1, the T cell receptor, is associated with CD8 T cells. You'll see there's one long chain and one short chain. So we can kind of see one long chain, one short chain here. Versus our T cell receptor is associated with CD4 T cells, have two equal length chains. And so we can kind of see this class 2 molecule, two equal length chains versus one long and one short. So that's the like biochemistry behind it. Really what I want you to think about is rule of 8 here. And I think I have this up in another slide, but if I don't, I'll mention it here now. MHC class 1 and MHC class 2 are the receptors that are going to interact with our T cells. And so or be associated with our T cells. So MHC class 1 is going to be the T cell receptor for our CD8 T cells. 1 times 8 is 8. This is terrible writing. MHC class 2 is associated with our CD4 T cells. 2 times 4 is 8. So when in doubt with MHC, it's your rule of 8. They should both always equal 8. So MHC1 is associated with CD8 killer T cells. So 1 times 8, 8. MHC2 associated with CD4 T cells, helper T cells. 2 times 4 is 8. So that's how we'll always separate the two. And we'll see here, these MHC 1s and 2s are going to be binding to different things and interacting. That's going to be part of what activates this T cell and gets this adaptive immune system process going. Both of these are presenting antigen fragments to T cells and binding our T cell receptors. So when we look at our HLC subtypes and our disease associations, we can start to see, okay, so our MHC different subtypes can actually be associated with different diseases. So people who have an HLC subtype of B27 could have potentially an upregulation or an increased association with these diseases from a genetic perspective. If they have the association with a DQ2 or 8, it could be associated with celiac disease. If we see, and these are HLA, sorry, let's see, HLA. If they're associated with DR3, these diseases, DR4, these diseases. And this HLA is referring to essentially the gene that's loci that's going to encode for these specific types of MHC complexes. So we all have a gene loci that's either like a HLA B27 or DR3 or DR2 or DR12. They're all different depending on our own genetic makeup. And then from there, we make our MHC1 and our MHC2 receptors. If you have one of these genetic loci or subtypes, you could have an increased risk for these certain conditions. And so that's where we can start to see some of that genetic interplay with our immune system. Even though it's not like the MHC caused anything, it's all based off of genetics and your genetic association with these diseases. But if you're struggling to kind of put those 2 and 2 together, these are the common HLA subtypes with the common diseases that they can have increased risk for.
Quick question.
Yeah.
So these HLA subtypes, I mean, all the diseases here are autoimmune. Is that always the case? If you have an aberrant genetic copy, it's going to be an autoimmune class of disease?
It's going to be an autoimmune class of disease. That's what they found thus far. They have not found a non-autoimmune class of disease that's been associated with these HLA subtypes. Yes.
Thank you.
Yeah, excellent. Absolutely. And so you can kind of separate them out here. This is a pneumonic pair. That's a mnemonic for these. So that's not a different disease. But yeah, so these are, I'd say 1 out of your 200 questions, right? Might be a question on this. So it may be low yield, but if it's something that you can stick in your brain, it does come up relatively often on NPLEX, at least once. So there'll probably be a question on something regarding that. Now, looking more at some specific cell types, now that we understand MHC, MHC class 1, MHC class 2, MHC1 associated with our CD8 T cells, MHC2 our CD4 T cells, and we'll understand more about T cells in a minute. I don't want to forget about our natural killer cells. They're a lymphocyte member of the innate immune system. They have a couple different actions. They contain porphyrin and granzymes, so they're able to induce apoptosis of either virally infected cells or really a cell that's infected internally. So if there's an intracellular bacteria, it would also affect that cell as well, or tumor cells. So anything that's intracellular, really, it can attack if it recognizes it as harmful to the body. It's induced to kill when exposed to some type of non-specific activation signal on the target cell, or if it has an absence of inhibitory signal. So the absence of an MHC1 on the target cell surface. If there's no MHC1 present, then it'll say, oh, well, this is not self, so I can kill it now. Because we'll see MHC1 later on is very non-specific, and so we find it on lots of different cell types that are our own, and that's how they're so good at presenting antigen to T cells. So if a natural killer cell comes across a cell that doesn't have an MHC, like over here, there's no MHC type 1, it's like, well, I'm gonna, get rid of this cell because there's nothing that's telling me that it's self anymore. You can also see an anti-dependent cell-mediated cytotoxicity that's essentially induced by CD16 binding a FC region of a bound IgG antibody. So an antibody is bound an antigen, or antibody is bound a actual cell, and the CD16 receptor on the natural killer cell binds to that FC region of the antibody, and we see that here. So that's how this could be activated. The natural killer cell is the CD16 receptor binds to the FC portion of an antibody on a cell that's been tagged for destruction. Natural killer cells will increase their activity with pro-inflammatory responses, so IL-2, 12, and interferon alpha and interferon beta. Our interferons we see especially upregulated with viral infections or with infections of something that's intracellular in nature, because our interferon gamma and interferon alpha and beta, but really all of our interferons are really stimulating a lot of things that can help target things that are inside of cells via apoptosis of some kind. So when you see interferons, think viral in general. You'll see these activating inhibitory receptors. So the natural killer cell function is the balance between the two. Activating receptors are the things that we've been talking about. There's those stress drive structures on cells versus inhibitory receptors are the MHC class 1 molecule. So if it sees that, it can be balanced even if it's seeing some activating factors. But if there's too many activating factors, it could override this inhibitory response. So looking at this again, we can see this is our natural killer cell. This is our normal cell here. We have an inhibitory receptor. We have the self class 1 MHC receptor here. They're binding together. So the cell's being inhibited, not being activated to kill. We can then see on this cell, there's no MHC class 1 present because it's a virus infected cell. So the MHC class 1 is gone. So the natural killer cell only has its activating receptor binding to the ligand. So it will be activated to actually apoptose and kill off this cell. A virus will typically inhibit class 1 MHC expression. So a cell that's infected by a virus will stop presenting MHC class 1 on its surface. And so that's a natural trigger then for the natural killer cell to say, oh, I can destroy you now. You're not like me. So when apoptosis is induced to infected or tumor cells, the killing mechanism is going to be the same as our cytotoxic T cells, our CD8 T cells. They use porphyrin and granzymes, and they're able to essentially apoptose that infected tumor cell or that infected or that tumor cell, and they bind together, kind of hold it in place using these two proteins, the FAS-L on the natural killer cell and the FAS protein on the cell that's being targeted for destruction. That's how it's able to actually stay communicated while it's doing its porphyrin, creating these pores, and then inserting these granzymes to break down and essentially digest the cell. Pretty grotesque, but pretty awesome at the same time too. Another important cell type that's involved with our adaptive immune system is our B cells, and our B cell development is an important process. B cells develop in the bone marrow. B cells are eventually they're cell focused at first, but really they're a part of our humoral immunity because they're gonna secrete our immunoglobulins, which are gonna be our antibodies. And the main action of our B cells or the main action of our antibodies is gonna be to recognize and present antigen. And so that's our goal of our B cells, is to get to the point where they are exposed to antigen, they become activated, and they produce antibodies. They also can maintain some immunologic memory. And so some B cells will become memory B cells that persist over time, and they'll increase our body's future response to antigens. And so they will not go off and produce antibodies and become a plasma cell. They will, in fact, just maintain their life as a memory B cell. Our T cells, I've mentioned a few times, our T cells maturing in the thymus will either transition over to become a CD4 T cell or a CD8 T cell, depending on kind of what components they are upregulating on their cell surface. CD4 T cells you could think of as helper T cells. They help our B cells to make antibodies and to produce cytokines to recruit more of our innate immune cells and activate other leukocytes. Versus our CD8 T cells are able to directly kill virus-infected and tumor cells, much like natural killer cells, using perforin and granzymes. When we have issues with our T cells, we can actually tie it back to a delayed cell-mediated hypersensitivity or a type 4 hypersensitivity, which we will look at in a moment. We'll look at our 4 different types of hypersensitivity. And this is that rule of 8 I mentioned. CD4 T cells interact with MHC2 molecules. 2 times 4 is 8. CD8 T cells interact with MHC1 molecules. 1 times 8 is 8. When we look at this activation, this is happening in a lymph node. We're zooming in and we're seeing how is the relationship between our antigen presentation of a T cell and its activation and antigen recognition by a B cell. So we can see here our T cell, our naive T cell in the parafollicular cortex, the T cell zone in a lymph node, is being exposed to a microbe, an antigen from a microbe via an antigen presenting cell. This could be a dendrite, this could be a macrophage, but it's presenting this via an MHC class 2 molecule to the naive CD4 T cell. So the CD4 T cell isn't specific for any antigen yet. Once it's been exposed to this antigen that's able to bind via this MHC class 2 molecule, it's then going to proliferate, divide, and differentiate into its effector CD4 T cell, its mature form. It's then going to migrate over where it's going to eventually interact with a B cell. And we can see over here, our B cell has also engaged and interacted directly with that microbe, that antigen. B cells are able to do that. And it started to create essentially these little antibodies. It's become activated. It's become specific for these antigens here, but it hasn't released them yet. These B cells then leave their follicular B cell zone, migrate into the T cell zone, and interact with this T cell. And the T cell, the helper T cell, will help the B cell decide, hey, what antibody should I be secreting or producing to respond to this specific antigen, this specific microbe or infection. So they work together before the B cell goes off and produces its antibodies to do so. And they'll only interact if they've been exposed to the same microbe or the same antigen. If this microbe was microbe A and this was microbe B, then they would not interact here. This T cell would find a B cell that's been exposed to microbe A. So they'll only interact here and bind if they've been exposed to the same microbe initially. All right. So far, okay. All right. So some hypersensitivity reactions. We have our 1, 2, 3, and 4, or A, B, C, and D. So anaphylactic or atopic, antibody-mediated, immune complex, or delayed. So our type 1 hypersensitivity is our anaphylactic or our atopic. This is our typical allergic response. It's fast. It could be, it doesn't have to be like a full anaphylactic allergy, but it can. It could be an atopic allergy, an ATP, like allergic rhinitis, right? It could be that type of allergy too. It's an allergen response. The immediate phase occurs within minutes. The antigen will cross-link with IgG antibodies on a mast cell that's already been pre-sensitized. It's ready and waiting to see this antigen because the body already knows it has a reaction to it. Once that mast cell binds with that antigen or allergen, it's going to degranulate and release all of its contents, which includes histamine, tryptase, and leukotriene. They are released into the surrounding tissue. That's going to start a response that starts that symptomatic response that we see where everything's like running and weeping, especially in like allergic rhinitis. And in cases of anaphylaxis, we see like an even bigger response where you can have, vasodilation and really big increases in fluid movement. And you can see your body's like full response, your respiratory system starts to close off because things get swollen. So obviously, depending on the allergy response, it can be really, really big and scary or more annoying. But it's the same basis. It's an IgE antibody that's getting activated on a mast cell, causing that degranulation. The late phase, then, hours into this reaction, you'll see chemokines are attracting more inflammatory cells, eosinophils specifically, and they are going to continue to bind to that allergen and continue to cause inflammation. And now they'll start causing tissue damage because they're releasing more of these kind of dangerous, granules out into the tissue space. So you'll start to see more symptoms occurring here. So when you think of type 1, think of your classic anaphylaxis or your classic allergies, your allergic asthma, allergic rhinitis. Any of those would be a good example of a type 1 hypersensitivity. Your main player is IgE antibodies with mast cells or your granular cells.
Dr. Petro,
Yeah.
this might be a question that's like for later. But with the type 1 hypersensitivity, is that then a Th2 response from?
Nope, not T cell. It would be it would be a misnomer to fully go down a T cell response because it's B cell mediated because it's antibody mediated. Now, are Th2 cells more involved with allergies? Absolutely. So when you're thinking of your helper cells, your Th2 are more involved in your allergy response in general or your extracellular response. So in this late phase, that's when you could maybe see Th2 cells coming into play. But for the majority of these hypersensitivity reactions, I want you to think of them as B cell mediated because they're antibody mediated. So I want you to take away T cells from the picture, at least from the onset, if that makes sense.
Okay, thanks.
Yeah, what the NPLEX often will ask is like, is this T cell or B cell mediated? And they'll try to trick you into saying it's a T cell mediated reaction. There's only one that's T cell mediated, and that's type 4. And so it's a good test question, but it confuses us a lot of the time. So that's why I'll keep T cells out of here for now, but we'll bring them back into the picture.
Okay, sounds good.
Yeah. When we look at our type 2 hypersensitivity, this is going to be B cell mediated and it's specifically antibody mediated. And all of these are antibody mediated, 1, 2, and 3, but this one like majorly, right? And it does so because the antibodies bind to the cell surface of the antigen or actually the extracellular matrix. So you'll see direct cellular destruction, inflammation, and dysfunction. The cellular destruction is because cells become opsonized by the antibodies, and that will lead to phagocytosis, activation of complement, and natural killer cell killing. So these are some examples of what that can look like from this type 2 hypersensitivity. So all of these would count as a type 2 hypersensitivity. We see like autoimmune hemolytic anemia, immune thrombocytopenia, even transfusion reactions and hemolytic disease of the newborn. These all can count within that category. The inflammation phase, you could see binding of antibodies directly to the cell surface. When antibodies bind directly to the cell surface, again, activation of complement, Fc-mediated inflammation. We see this with Goodpasture syndrome, rheumatic fever, and transplant rejection. So full-on rejection of transplantation. So like graft rejection here. And then cellular dysfunction, antibodies bind to cell surface. You'll see blockage or activation of other downstream inflammatory effects. So myasthenia gravis, Graves' disease, and pemphigus vulgaris. All of these count as a type 2 hypersensitivity. So an antibody mediated. The kicker, antibodies are binding to the cell surface. That's the kicker. Versus we'll see here, antibodies binding to a released antigen and mucking up the system elsewhere. These are directly because antibodies bound to a cell involved in these different conditions or pathologies. And now those cells are being targeted and attacked. So it's a type 2 hypersensitivity. All right. Now, our type 3 hypersensitivity. These are our antibody antigen immune complexes. So we'll see antigens and antibodies, typically IgG involved. This was also IgG typically, but these antigen antibody complexes activate complement, part of our innate immune system. This is going to attract neutrophils, release lysosomal enzymes, and wherever these antibody antigen complexes go, we're going to see immune destruction follow. So that's why we can see these in areas that are affecting a lot of times smaller places of the body, like the kidneys. So systemic lupus erythematosus, we can see it with our post-strep glomerulonephritis, IgA vasculitis, polyarteritis nodosa, RA, reactive arthritis. But your classic example would be your post-strep glomerulonephritis. That's your like classic example of a type 3 hypersensitivity. You have an antigen, an antibody, they bound together, they get stuck essentially on that basement membrane of the filtration membrane of your glomerulus. And then we're bringing complement neutrophils, lysozymes, and they're attacking those complexes. Well, but they're also attacking the tissue around. So you have a disruption or destruction to that glomerular membrane, and then you're going to see issues of filtration, GFR, kidney function. The clinical characteristics, typically it'll be after a couple weeks of antigen exposure, enough time to produce those antibodies and create those complexes. You'll see fever, itchy skin, painful joints, protein in the urine, that's from the breakdown of the membrane, and lymphadenopathy. And then there's two kind of sicknesses that you can have: serum sickness, where complexes will deposit into tissues, like I've mentioned, or an arthritis reaction, where you'll have a local kind of subacute immune reaction. This happens with like a booster vaccine, local edema, fibrinoid necrosis, complement activation. That's typically just like right where that injection site was, and then it will resolve. So that's typically where you'll see it there. And then our last type of hypersensitivity is our type 4 hypersensitivity. And this is our delayed hypersensitivity. And there's kind of two mechanisms, but again, these are T cell mediated. B cells are not involved at all in this type of hypersensitivity. One is direct cell cytotoxicity. So our CD8 cytotoxic T cells will kill off targeted cells. And the other is our inflammatory reaction. Our effector CD4 T cells recognize antigen, release inflammation-inducing cytokines. It's kind of a slow burn. There is no antibody involvement in this type 4 hypersensitivity, or this delayed hypersensitivity, which is why it's slower in onset. And a classic would be like contact dermatitis or graft versus host disease. Those would be your classic picture of type 4 cell-mediated hypersensitivity. And the kicker, cell-mediated, T cells, not our antibodies. All the others were antibodies. Okay. If you're like, I don't know, that was a lot, give me a table. There you go. Here's your table. So anaphylactic, cytotoxic, immune complex, delayed type, showing antibody involvement, the antigen response time, appearance, histology, transferred with, and some examples. Hey, all right, let me pop out of this to see your faces, or at least see the pictures that you have for your faces. What else?